All right, so we're moving on from DNA and DNA replication and going into protein synthesis. So you do not need DNA replication to occur to get to protein synthesis. You do need DNA, however. So the processes are going to be a little bit different from one another, but we are going to rely upon DNA as well as RNA now. So remember, DNA is found in your nucleus. It makes up your chromosomes. It's double-stranded. It's huge. It's a very large molecule. It's too large to leave the nucleus. However, in order to make proteins, we have to make them out into the cytoplasm. So we're going to go through this process of protein synthesis. It is a two-step process, transcription and translation. So when we talk about transcription and translation, replication is completely different. I don't need replication to occur in order to have transcription and translation. I just need the DNA molecule so I can get the code within that, the A's and T's and C's and G's and how they're put together in order to go out and make my proteins. So we're going to break this two-step process down. Today we're going to look at the first step, and we're going to go through that just a little bit, and then we'll talk about translation from there. So once again, I'm going to kind of be backtracking between notes and the coloring page that you have. There's one page within the coloring packet, and then there's a little kind of like fact sheet that you're going to see in color that goes through just protein synthesis for you. So your first phase of protein synthesis is called transcription. You are transcribing the DNA into a form that is smaller and is able to go out into the cytoplasm. So this first phase of protein synthesis is going to start out in the nucleus because we need to take DNA's code from the nucleus to the cytoplasm. So before we get there, we know that we're relating it or need to worry with RNA. So we got to talk a little bit about RNA and how it's different than DNA. RNA is ribonucleic acid. Remember, DNA is deoxyribonucleic acid. It is still made up of nucleotides, which are still one sugar, one phosphate, one nitrogen base, but there are some differences between the DNA nucleotide and an RNA nucleotide, and the DNA strand and RNA strand. So first off, DNA is big, it's double-stranded. So two strands of those nucleotides, a double helix. RNA is single-stranded, it's much smaller. That's the reason it can fit through the nuclear pores. That's the reason it starts out in the nucleus and works its way out to the cytoplasm because it is so much smaller. Our sugars are different. In DNA, you have deoxyribose. In RNA, you have the sugar ribose. They are both 5-carbon sugars. It's kind of like they're isomers of each other, kind of like glucose and galactose that are 6-carbon sugars. And then all of your base pairs are the same except for one. So in DNA, we are replacing the nitrogen base pair thymine with uracil. Uracil did everything that thymine did in DNA. So when we get to this RNA, uracil is also a pyrimidine. It's a single ring structure, just like thymine was. It's going to bond to adenine, just like thymine did. It's going to join with 2 hydrogen bonds, just like thymine did. So it's just filling in for thymine when we get to our RNAs. So you have 3 types of RNA. Messenger RNA, we'll talk about transfer RNA, and we'll talk about ribosomal RNA. And they're all going to kind of be involved in one step or another, or both, of protein synthesis. All of your RNAs are made within the nucleus, and they kind of travel out to do their job where they're needed. So your transfer RNAs are going to travel out to the cytoplasm. Your ribosomal RNA is going to travel out to the cytoplasm. Messenger RNA is going to be created within that nucleus and then travel out once it gets the DNA's code, which is what its job is. So mRNA is a single uncoiled strand. We need to take mRNA and copy DNA into messenger RNA's code. What does that mean? It means that where we have thymine, we're now going to put a uracil in it. We're only copying a section of it. We're not going to do the entire chromosome. We want to copy a gene. We want to make a protein. It's a segment of your nucleotides or a segment of those nitrogen bases. It might be 50 base pairs. It might be 150 base pairs. It might be thousands of base pairs long. And that amount of base pairs is going to code for a specific protein trait that we need to get the code from the nucleus out to the cytoplasm. So messenger RNA is going to come in and do that. So it's going to bring the DNA information because we're going to transcribe it into messenger RNA from the nucleus and take it out to the cytoplasm to a ribosome. This is the template or the pattern that helps us to assemble together our amino acids during protein synthesis. Because if you remember back to organic, we said proteins are made up of our building blocks, which are amino acids. 20 different amino acids. The way that we string them together make unlimited number of proteins. Just like in DNA, we talked about every three base pairs, AAA, ATT, GCC, whatever it may be. Every three base pairs in DNA was known as a triplet. Here, those three base pairs are now going to be known as a codon. So codons are three base pairs in messenger RNA. It's this codon or three base pairs in messenger RNA that are going to code for your amino acid. So first your bio may have looked like a big codon wheel and had letters of A's and C's and G's and U's. Now on the outside it had all 20 of your amino acids. You guys are going to see that chart again. We'll talk about that one. There's also a square one that we're going to look at, and that's going to allow you to determine what amino acids are we stringing together to make our particular protein. So messenger RNA, once again, mRNA, is taking the DNA's code, putting it into a smaller single-stranded version of it, and then shipping it out to the cytoplasm, where we can hook up to a ribosome for this entire process to begin. Transfer RNA, your tRNA, they're kind of like little taxi cabs, I call them. Basically, they are carrying your amino acids out in your cytoplasm. So once again, they're made within the nucleus. They go out, they travel out into the cytoplasm, and they pick up a specific amino acid. Remember, there are 20 different ones. So he can't just go and go, hey, let me pick up this one. Let me pick up methionine this time. Let me pick up adenine this time. He can only pick up one specific amino acid. And he knows which one he is going to pick up based on his three base pair code that he has on the bottom of them, of U's and A's and C's and G's. This three base pair code on tRNA is called an anticodon. So what we're going to see here in a moment is that your anticodon on tRNA and your codon on messenger RNA are going to be complementary to each other. So if this was going to match up to part of my messenger RNA strand, I would say, oh, U is going to match up to A, G is going to match up to C, G matches up to C. Anticodon will match with codon. And that allows us to bring in a specific amino acid. But that's going to be more translation when we get there. The transfer RNAs kind of look like little clover leaves, so they're kind of wrapped around these little base pairs here, but the base pairs at the bottom, once again, every three base pairs is an anticodon. They can only, once again, bond to one specific type of amino acid that's floating out in the cytoplasm. Once again, these amino acids are coming from the foods that you eat. So you're not eating a balanced diet and you don't have all those 20 different amino acids within your system. You can't just pick up anything. So if there's not an amino acid there for him, he doesn't have a job to do. The one that we usually forget about is your ribosomal RNA. So ribosomal RNA, or rRNA, hooks together and activates your ribosome. So you think of these little ribosomes as teeny tiny little balls out in the cytoplasm. They can either be free floating in the cytoplasm or they can be attached to the rough endoplasmic reticulum. So it kind of depends upon what your body's going to do with that particular protein. Are we using it inside this particular cell or are we shipping it out? And that determines whether it's made free floating ribosomes, or are we made within the endoplasmic reticulum and we're used within the cell? So when we get to translation, we'll look at some of those differences a little bit more. But your ribosome is just not a little ball. It's actually a big subunit and a little subunit that need to be kind of joined together and activated. And what's going to do this is that rRNA. This activation kind of makes it like a decoder ring. So now when this messenger RNA strand goes through my little ribosome and gets pushed through it, he's able to read each codon. So the little picture you guys see here above, the blue is my DNA. A couple ways you guys should know that. First off, it's double-stranded. Second off, it has thymines in it. So when we go through and we transcribe DNA into messenger RNA, I'm only doing a small region of it. So we're going to have kind of this initiation section or a promoter region that says, hey, this is where the beginning of the gene starts, the code for it. That DNA is just going to break those hydrogen bonds. And instead of DNA polymerase, which is how we made new DNA, RNA polymerase is now going to come in. And it's going to read the one side of the DNA that the code is located on. And it's going to match up base pairs. So it goes along and it goes, oh, here is an adenine. Let's put uracil. Remember, there is no thymine in any of our RNAs. Here's thymine in DNA. I still have adenine in RNA. Cytosine goes with guanine. Guanine goes with cytosine. Adenine goes with uracil. And it keeps working in that direction. So just like DNA, and this is the good part, DNA still gets read 3' to 5'. Messenger RNA still gets made 5 to 3. So exactly like your DNA. DNA is read 3' to 5', it gets made 5 to 3. When we go through transcription, DNA is read 3' to 5', messenger RNA is made 5 to 3. And you would determine that end the same. You would find your sugar, locate your oxygen, count your carbons to know which end is which if one is not labeled. So let's look at our process now that we have messenger RNA, transfer RNA, and ribosomal RNA. So this particular process, we just looked at that picture just a little bit and we said, well, that DNA strand is just going to kind of unzip a little bit. We're going to break those hydrogen bonds, and helicase is still involved with that particular breaking of those hydrogen bonds. It's only doing a small section there. It could be thousands of base pairs, it could be hundreds of base pairs. So as that DNA unzips, messenger RNA bases, you have RNA nucleotides free floating in your cytoplasm as well, or I'm sorry, within your nucleus as well, which is where this process is taking place. These nucleotides are being added. So as RNA polymerase is reading the old DNA strand, new messenger RNA nucleotides are coming in and matching up. So that's the enzyme that's responsible for this polymerization, this production of our new messenger RNA during this process of transcription. Eventually, just like we started with this initiation or promoter region, it's going to basically say, hey, this is the end of my particular segment that I need to copy, so let's stop here. Here's the chain that I have. Now I'm going to have this single strand of messenger RNA. Messenger RNA then breaks off from DNA. DNA just goes right back together again. Those two strands, hydrogen bonds come right back together again. I don't need to go through and replicate or anything else, different processes. So once I've transcribed that little section, my original DNA, the hydrogen bonds between those DNA base pairs come right back together again. My single strand of messenger RNA is going to exit the nucleus. It goes through one of those little nuclear pores that DNA is too big to get through. It's then going to go out into the cytoplasm and it's going to hook up to a ribosome. That's where that second part is going to take place for us. So, just like we said before, messenger RNA is synthesized just like DNA. It is read, the old DNA, 3' to 5'. Messenger RNA is made 5 to 3'. So if we take a quick little look at our coloring packet, the same as what we had before, we're going to say this looks just like the previous picture, and it absolutely does. It's still taking place in the nucleus. It is transcription, first of two processes that we see with protein synthesis. So I go, oh, replication is DNA being made into DNA. Transcription is DNA being made into messenger RNA. Anything with DNA has to take place in the nucleus. So replication takes place in the nucleus, transcription takes place in the nucleus. At the top, it gives you guys your differences. So between DNA and RNA, first off, my sugar is different. Second off, my base pairs are different. Uracil does everything that thymine did. So you can see that here are my little pyrimidines. They're both single ring structures. Here and here. Here are my sugars for ribose and deoxyribose. They're isomers of each other. DNA is double-stranded. RNA is single-stranded. So there are your differences between it. This double-stranded DNA, it's going to temporarily unwind only a segment of it. Polymerase is going to come in. It's going to read my old DNA strand from 3 to 5'. It's going to make my new DNA 5' to 3'. Remember, every 3 base pairs in DNA is called a triplet. So here I can go TAC. That's a triplet. That triplet is complementary to codons in messenger RNA. My codon messenger RNA would be A, U, G. T goes with A goes with U, C goes with G. My next triplet, G, C, A, is complementary to my codon C, G, U. Guanine goes with cytosine, cytosine goes with guanine, adenine goes with uracil, and it would keep going all the way down. Eventually, we hit that stop region. This single strand breaks off, exits out the nuclear pores. My original DNA starts bonding right back together again, those hydrogen bonds. The last little sheet up there is a fact sheet, and I found this one online, and I really like this particular one because it does break them down a little bit more for you guys. So we said, okay, well, here is my double-stranded DNA. I'm only going to take a part of it, and that region is called the promoter region, and that's where RNA polymerase is going to attach to. And let me attach here. This is where we're going to start copying that particular gene, that segment of DNA that's going to code for protein or trait. Helicase will still come in and kind of break that open, kind of make that little fork-like area for us, and it's going to expose those particular nucleotides so that we can go through and copy them into a language that messenger RNA understands. It's smaller, it's single-stranded. It replaces thymine with uracil. It's in its own language. It's transcribing the DNA language into a language that messenger RNA is going to understand. So as we go through, messenger RNA pairs with the DNA template. That DNA strand that we're making a copy of or a complement of base pairs from is called the template or model or mold. So guanine goes to cytosine, adenine to thymine in DNA, but now we have, adenine in messenger RNA will pair with thymine in DNA. We have uracil in messenger RNA that's going to pair with adenine. So in DNA, adenine, thymine, guanine, cytosine, you will never have thymine in any of your RNAs, transfer RNA or messenger RNA. We keep adding them. The nucleotides are added until the polymerase reaches a termination sequence. It says, hey, this is the end of the gene. This is all I really need to transcribe into my language. I'm done. DNA kind of gets released. The polymerase gets released from that, the enzyme. Original DNA comes back together again. And you can see here, this was my nucleus. Messenger RNA moves out through the nuclear pores and goes out into the cytoplasm. It's out in the cytoplasm that this whole process now takes place. So remember, every 3 base pairs, he's got the code for proteins, for any trait that we need to do, anything that we need to create. This is the code, the building blocks on how to put these amino acids together to make that particular protein. And that's what the second part of protein synthesis is going to deal with. And that's going to deal with your translation that we're going to see. So that's going to be the next part that we're going to kind of let that sink in. We'll talk about translation next time we see each other.